Okay, so we went over how bacteriophages, kind of attach, replicate, and release from a cell. Um, we're going to talk a little bit about animal viruses and how it works in animal viruses. You'll remember that in addition to the capsid in the genetic material, most animal viruses also have a viral envelope that is derived from the host cell membrane. Um, Overall, animal viruses are more complex than bacteriophages. Um, but that first stage attachment is fairly similar. In this case, a spike protein, so on the outside of the virus, is going to bind to a viral receptor on the host cell. And this is similar to the bacteriophages, where this is kind of a lock and key type mechanism. The spike proteins are very specialized for a given viral receptor. And again, that receptor on the host is is evolved for a different reason. It hasn't evolved for the virus to attach to. It has some other function with the host, but it's kind of taken advantage. The virus takes advantage of it, and uses it to attach and then gain entry. Again, also like bacteriophages, this is very specific. And so not only are these going to be kind of species specific in terms of what what types of species the viruses can infect, but it also is going to be specific to kind of certain cell types as well that have the receptors that they can bind to. All right, so after attachment, we can see entry, and we can look at two different means of entry. So one is fusion of the viral envelope in the plasma membrane. So after receptor binding, the virus will, that viral envelope will fuse with the plasma membrane of the host, and the inside of it, so the capsid in the genome, are going to enter the host cell. That viral envelope will kind of remain joint with the cell membrane. Another type is endocytosis, and so after the binding to the cell receptor, this is going to trigger the host cell to kind of engulf the virus, the viral particles, um, and bring it into the cell. And I'll note that most most animal viruses do have that viral envelope. Um, but if they don't have the viral envelope, they cannot enter through fusion, right? There's no viral envelope to fuse with the plasma membrane, and they would be entering through endocytosis. Okay. Um, and so for replication, there's two things that need to occur, right? So synthesis of it, the capsids, so the proteins that are going to make up the capsid, or the outer portion of the virus, needs to make more copies of that, and it needs to make more copies of its genome, so the viral genome. How it goes about that is really dependent on what type of genome it has. So is it made up of DNA or RNA? Is that double-stranded or single-stranded? So that's going to make a difference in how, kind of what steps are needed for the translation of the viral genome into capsomeres, and the kind of copying of the viral genome to be part of the new viruses that are made. And so here it mentions any viral enzyme. So dependent on the type of genome, sometimes viral enzymes need to be made and actually carried around with the new viruses. And so if you aren't very familiar with the steps of just DNA replication or the central dogma of, DNA being transcribed into messenger RNA and then that being translated into proteins, make sure to do a little refresher on how that works. You don't need a lot of details for understanding this, but you do need to know the basic ideas. Okay, so all viruses need to recreate their original genome type, right? So they need to make more copies, replications of their genome for the new viruses. And they all need to end up making messenger RNA, which is the same as the positive single-stranded RNA. And this is necessary to make any of their viral proteins, right? So for translation to occur, it needs mRNA, that messenger RNA. And so some types need to carry enzymes around to actually accomplish this. They can't just show up at a host cell and be able to do this because not all the enzymes that the virus needs are available in the host, and I'll show you what that means in a moment. Okay, and so I'll walk through these fairly briefly, but I will point out that if the viral genome is the positive single-stranded RNA, that's the same as mRNA, and that is directly translated into the viral proteins. And so that's a pretty straightforward one, at least for that translation. Okay, so viruses with DNA genomes first with double-sided or double-stranded DNA. These steps are going to be really similar to DNA replication, transcription, and translation, right? So we have double-stranded DNA. It's replicated within our cells, and that double-stranded DNA is used for transcription to messenger RNA, which is then translated. And so these steps are all basically the same as what's going on in animal cells already. They're just using the viral genome to do it. If we look at single-stranded, in this case, the positive single-stranded DNA, this is very similar in how it all works, but there's just this one additional step. So it starts with single-stranded DNA. It's going to make the complementary strand. So then you end up with double-stranded DNA, then the same steps as above. All right, when we get into RNA genomes, it becomes a little more complicated. So in our cells and animal cells, we basically go from DNA to RNA. We don't work in the other direction, and we're usually not copying RNA from RNA. And so animal cells don't have kind of the equipment that the viruses need for replication. So if we already mentioned the positive single-strand RNA is the same as messenger RNA. So this one is very easy to just go right to translation. And one of the proteins that it's going to make during translation, in addition to the capsids, is replicase. Replicase is an enzyme, also known as RNA-dependent RNA polymerase. right? So it's RNA dependent, so that means that it's using template RNA. And then RNA polymerase. So polymerase is an enzyme that puts RNA together, right? So replicase uses template RNA to synthesize new RNA. And so that's what with the positive single-stranded RNA, the translation goes real smoothly, but it needs to make more copies of itself. It needs to replicate its genome. And animal cells don't have replicase. They don't have a polymerase to make more RNA from RNA. And so it will, after the replicase has been made, it can then use replicase to make a complementary strand, so the negative single-stranded RNA, and then use replicase yet again to make to use this complementary strand as a template and make more of the positive single-stranded RNA. All right, and so this is used for the new copies of the new copies of the viruses as the genome. Single-stranded or negative single-stranded RNA, it's a little bit trickier. So it cannot, give me one. Okay, I need to pause this for just one moment. Okay, so this becomes a little more complicated because the negative single-stranded RNA cannot be directly translated, right? But in order to kind of make the complementary positive single-stranded RNA from the negative, you need replicase. And so there's a little bit of a problem here where it can't make the replicase because it needs the replicase before it can express any of its genes. And so it kind of solves this by bringing replicase with, right? So when it was created in a previous host cell and assembled into like this viral particle, it included replicase. So it brings its own replicase with. It uses that replicase to make a complementary strand, so that positive. Now this can be used for translation, and it can make more replicase along with other capsid parts. And then this complementary strand that it made, it can be also be used with replicase to make more copies of the negative strand. And so so when it kind of is all packaged up, it's bringing the replicase with it when it leaves the host cell. And so that's different than the positive ssRNA, which can, when it gets to a new host, it can make replicase for itself right away.